Okay, well, yeah, thank you for the introduction. So as I say, I'm a postdoctoral research fellow in the University of Leeds. And I mean, you have summarized my history. Just want to mention that I guess the way I also came into work with tribology, the same as professor, is just by chance because I mean, my background, it is in food science, but I'm a colloidal scientist, but of course it's related. So I just had the opportunity to enter this space, and now I'm fully in, I guess. So anyway, I'm just going to share a little bit of what is like oral tribology and just an example of the research that I've done, specifically applied for the design of food matrices for all the population. I'm not going to go in through the whole details of everything because we don't have time for that, but just an example of the work that I've done and some of the results that I found and some of the challenges as well that I have encountered. But just a little bit as an introduction for, I mean, whoever is not familiarized of what is oral processing or tribology and so on. So, I mean, we know that eating is a dynamic process. And because of this, studying kind of like the sequence of oral manipulation beyond the first bite to use it for rational food design, it's very challenging. So one of the approaches that has been used in order to kind of like develop these in vitro techniques that seek to like capture what the food experiences is during the oral manipulation has been to divide the oral process into different stages to try to identify like which texture is perceived during which state of the oral processing. So I should note also that in reality, these processes actually like overlap, but studying them like separately allows to understand kind of like the underlying physics to get some insights into the specific like functionality of food ingredients or different like food components. So yeah, I mean, so for the purpose of developing in vitro approaches that enable like this rational design of food, oral processing has been split into six different stages. So the first one being like the first bite, and then the comminution or the chewing, then granulation of the food, and then the bolus formation, of course, swallowing, and then whatever residue is left in our mouth. So through this model, we can see that there is a transformation from a rheology-dominated process, so during the first bite, to a tribology, dominated process during the oral processing, since there's a lot of surfaces interactions in the mouth between the food particles and the different oral surfaces in our mouth, such as our tongue, our palate, our teeth, and so on. So tribology becomes like very important. So what is then oral tribology? So oral tribology is the study of friction, lubrication, and wear of materials in the oral cavity, particularly focusing on the interactions between food and other substances and surfaces like the teeth, the gum, the tongue, the palate, and so on. And it involves understanding how these interactions affect oral health, sensory perception as well, and oral comfort during activities such as chewing and swallowing. And oral tribology like plays a crucial role in food design and sensory perception by providing kind of like some insights into the mechanical aspects of eating and kind of like guiding the formulation of foods that offer like optimal texture and flavor release or swallowing comfort as well, and some nutritional benefits. So during oral processing, we encounter kind of like the three regimes like normally found during tribological processes, with of course the dominant regime varying based on the food type and the processing stage that we are in. So with, our already well-known Striver curve. Kind of like the lubrication behavior of food samples can be understood depending on the different amount of food that it's between the rubbing surfaces. So, for example, the hydrodynamic regime is generally considered to be related to the start of the oral process when the food is consumed. Whereas in the case of the mixed regime, it's when, we have some entrainment of food between the surfaces, or in this case, the tongue and the palate, and it's partially separated by the food. And finally, well, in the case of the boundary regime, it's essentially when we have like some residues of food, or what we call like the boundary film. And well, this boundary lubrication is characterized, of course, by the presence of like a layer of residue in the tongue and the palate that does not participate in, the hydrodynamic flow of the bulk food. And I mean, in this regime is where the kind of like the two surfaces that we have will come in contact, and the asperities like support the load. And it's normally closely related to the human perception of astringency and slipperiness as well. So, for example, a very good example of a boundary lubricant is actually saliva. So the proteins in the saliva absorb to the surfaces or to the substrate to form like a multi-layer film that protects the oral surfaces from wear and friction. So for the study of oral tribology, there are like different equipments, but the ones that we use in Leeds and the ones that I have a bit more experience, it's the MTM, or the microtraction machine. And we use it essentially because it's quite versatile, and it allowed us to test in a wide range of entrainment speed. I think it goes all the way to like 3,000 millimeters per second. But we also have a rheometer that has different tribological setups that we use at different surfaces as well. And one thing that I guess is important to emphasize is that, well, it's important to remember that friction is a system property. So meaning that, like the surfaces that we use, or the different parameters that we use, such as the sliding speeds or the temperatures or the geometries that we use, are going to affect the results that we have. So, the results that we're going to have from one equipment, so from the MTM to the rheometer, are going to be completely different. So another thing that is very important to consider when we study oral tribology is, of course, the role of saliva. So essentially, there's kind of like three main interactions between the food and saliva that can lead to three different outcomes. So either saliva can lead to either minimal interactions, where essentially the saliva film lubricating properties remain like undisturbed, or it can lead also to like a disruption of the salivary film, causing like an increase in the friction. I mean, potentially due to like changes on the pH or ionic strength, or like protein aggregation that it's induced by some different food compounds, such as like polyphenols. Or it can also be, there can also be a synergistic interactions where certain food compounds can alter the properties of the saliva, which affects, the friction and, the mouthfeel and the texture of the food that we perceive. And well, these interactions, of course, plays a crucial role in understanding how different foods are perceived and experienced during consumption. So now I'm gonna talk a little bit about what I've done using oral tribology. This is a project that I've been working for the last couple of years. It's called Eat4H. I mean, it's called palatable, nutritious, and digestible food for the prevention of undernutrition of active aging. And well, essentially the reason why this project came out to life, kind of, I mean, because it's not a secret that aging affects the way we eat, with the loss of appetite actually being the major cause of undernutrition in other adults. So there are multiple factors that might be involved in these, for example, like the decline of the sensory perception, or the decline of the quality of the saliva, or the decline even on the dental health that we have, or the tongue strength that we have, that could mean that eating food becomes like a real challenge, as you know, it's hard to chew, or it's hard to, if the quality of the saliva is not good enough, it's hard to like humidify the food, or it's even hard to swallow the food, which causes a lot of oral discomfort, and of course like poor health. And, you know, there's also other factors such as depression and chronic disease, or even like polymedication, that also play an important role in the loss of appetite in the aging population. And all these eight related changes can influence the oral processing, and of course the sensory experience that we have. So the idea of designing like food for vulnerable populations is that the texture can be potentially modified to enhance oral comfort, increasing hopefully like food intake in older adults, which, I mean, we know that it's important for life quality, life expectancy, and overall like life enjoyment as well. So the aim of the project is to provide novel insights on designing food products with kind of like just right textural properties to enhance consumer acceptability. We are focusing now on 3 different food categories. So we have a dairy category where we are working with a soft cheese and a yogurt. And we also working with a product that's called dulce de leche, which is something similar to like the salted caramel. We're also focusing on a meat product, and also a cereal product that is similar to the cereal, I think it's called Crave from Kellogg's. So, I mean, the idea of these products, it's to be like fortified with different proteins and bioactive compounds to, as I said, like tackle undernutrition in other adults, and also to impair the loss of muscle mass, which is also quite important in other adults, because it's very common that if you lose the muscle mass, you're more prone to like fall and break bones, or you know, these kind of things. The problem is that when you design or you enrich food products, especially with proteins, with other adults, you start creating, well, you promote the sensation of astringency, which enhances the mouth drying perception, and this tends to drive like rejection from fortified food in other adults. And although this exact mechanism of astringency and mouth drying is not like completely understood, it has been observed that, well, it can be caused by like polyphenols, like interacting with the proteins that we have, like mucoproteins in our mouth, which leads to like increasing friction. So we are hoping that with this project and by using our tribology, we might be able to understand a little bit more, or if possible, to relate if there's some specific ingredients in these food products that we're using that might be promoting like increasing friction coefficient, and if this knowledge can help us predict or see a correlation between the sensory perception of these products to hopefully use it to design future products. I'm not going to go through all the data of all these products because I don't have time. But I'm going to present the data, for example, for the dulce de leche. So in this case, we had 3 samples. So the first one was the control sample, which was a regular recipe to make this product. The second sample that we had was a product that was enriched with whey protein isolate. And the third one was a control that was enriched with whey protein isolate and also with hydrolyzed whey protein. So we characterize them in Leeds using different techniques such as particle size, rheology, confocal, tribology, of course, and QCMD. And also, since this is sorry, I forgot to mention, this is a collaborative project with different universities in Europe. We're also collaborating with the research institutes in France, in Norway, in Israel, and I'm missing Ireland as well. So we have also done, well, our partners in Norway, they have done the sensory analyses, and our partners in France, they've done some oral comfort and salivary addition studies as well. So I'm going to present just some results of this. Okay, so for the first analysis that we've done, like simple characterization of how it looks and, you know, the particle size of these samples, we essentially saw that they have significant difference in the size and the appearance of the samples, which is very evident that especially the samples that were enriched with whey protein isolate and hydrolyzed whey protein, you could see like some protein aggregates there. It's quite visible compared to the control samples. In terms of the rheological characterizations, I mean, they all present like shear thinning behavior with some samples being less shear thinning, such as the control sample, and the ones that it's enriched with whey protein isolate and hydrolyzed whey protein having a stronger shear thinning behavior. I'm not going to go into the details, but saying especially at the lubrication properties of the friction measurements of these 3 samples, we could see that, for example, the control sample and the one that was enriched with the whey protein isolate kind of like display a hydrodynamic lubrication regime across the entire, almost the entire speed range that we use. Whereas we can see that the one that was enriched with whey protein isolate and hydrolyzed whey protein, it kind of presents a little bit of a boundary, I guess, and a short mix regime, and mostly a hydrodynamic regime as well. So there is a difference between, especially for the one that has the whey protein isolate and the hydrolyzed whey protein. Then we have also done some QCMD analysis. So for those of you that are not like familiar as with what it is, essentially it's just some absorption measurements into hydrophobic surfaces that resembles the hydrophobic nature of the mouth, well, of the tongue. And we are able to see, for example, how much mass is absorbed of, in this case, these 2 proteins that we are using. And what we notice is that in the case of the whey protein isolate, we can see that it absorbs more into the surfaces, but after we rinse it with some buffer, we can see that the rate of desorption is way bigger than the hydrolyzed whey protein one. And also, I mean, using different models, we can also see the nature of the film. So we can see that in the case of hydrolyzed whey protein, it's forming a very rigid film in the surface. What we did later was to incorporate some model saliva to try to see if we can mimic it, like the in vitro bolus formation of the sample, and we use the same amount of saliva that was observed from our partners in France that is added to this kind of products. And what we could see that first is that, well, there was no changes in terms of the size and the appearance when we added the model saliva. There was not a significant effect on this. And that actually, in terms of the lubrication and of course the rheology, we could see that actually the model saliva had more of a diluting effect, where the diluted sample presented, well, of course, lower viscosity, which in turns this translated into the fact that because of the lower viscosity, there was a decrease in the ability to support the load during the tribological measurements. And we can see that the friction essentially just increases in all of the three samples, and there was no significant difference between them. Then if we consider the sensory analysis results from the sample, what we can observe is that in the case of the control sample, they had a relatively high textural intensity of like firmness, fullness, and adhesiveness. Whereas for the samples with enriched with a whey protein isolate, there was a relatively high intensity, textural intensity on the granularity. The same with the samples that were enriched with both the whey protein isolate and the hydrolyzed whey protein. There was a relatively high intensity of granularity, of astringency, and also like aftertaste. And although we are currently actually working with a statistician on to see if we can correlate the tribological measurements that we done and the sensory data that we have. So I still don't know if it correlates or not, but I guess like from the results, it's really interesting to see that it appears that that there's some relationship, especially for the sample that was enriched with whey protein isolate and hydrolyzed whey protein, that it's perceived as more stringent and more granular compared to the other samples, which it might be related, again, I still cannot say for sure, but it might be related to how it behaves in the tribometer, and also to the film properties that it has, meaning that it forms more of a rigid and it binds more strongly into hydrophobic surface beds. So it's going to be interesting to see if there's, some correlation here. And then, I mean, just to finalize this, I've just added some conclusions of basically, it's a bit of what I learned so far. So, I mean, we know that tribology is a system property. It's not an inherent material property, which kind of like emphasize the importance of choosing the right instrument with the right surfaces, and also specifically based on the food matrix that you're using. So in this case, I mean, it was quite easy to analyze the dulce de leche because it's a liquidish thing, but we are also working, as I said, with like solid samples. That has been quite challenging. So Also kind of like I learned that, bridging the gap between in vitro tribological measurements and in vivo oral processes and the sensory perception, it requires a better understanding of the entire system, including, the surfaces that we use, the equipment and the food matrix, the state of the food matrix. And well, as friction measurements are system dependent, kind of like all the in vitro techniques describes have their limitation. Also, well, it's been quite challenging to correlate friction with the sensory characteristics. So that remains quite complex and there's a lot of potential there. But the good thing is, as the field of food tribology or tribology continues to expand and there's interest in this, I feel that this work done, it's opening the doors towards like a better understanding of tribology in relation to like oral processing and sensory perception and how we can use these for like rational food design. And well, I just want to thank, of course, my, former supervisor and now boss, Professor Nweshe Sarkar, and also Alan Mackey, and of course, all my colleagues in Leeds in the School of Food Science. Thank you.